Finding out that the aging benefits in these little critters are derived from the same NRF2 genetic pathway that is conserved in humans is really good news if we're ever to have any hope in replicating this data in a species a little more closer to us. Finding out that the FOXO genetic pathway is also involved is really just a cherry on top. The FOXO3 genetic pathway, which I've talked about in multiple podcasts and videos, is very closely related to human longevity. For example, humans that have a gene polymorphism in the FOXO3 gene that make it more active are 2.7 times more likely to live to be a centenarian. The chances of living to be a centenarian, however, seem to depend a lot on keeping inflammation at bay. In fact, it is now believed that suppression of inflammation is the single most important predictor of successful aging. And we're not just talking about lifespan either. We're talking about capability in terms of performing functions of daily life, and also cognition. Yes, inflammation was an important predictor of cognition itself, and this was shown in populations including elderly people all the way up through super centenarians, which live to be 110 years old. And this relationship isn't surprising. We know that if we take mice, for example, and induce chronic low-level activation of a master regulator of the pro-inflammatory response, known as NFkB, it can actually accelerate aging by 30% in mice. Again, suggesting that chronic enhancement of pro-inflammatory mediators really is not just a bystander, but an actual driver of aging. Sulforaphane has been shown in mice to inhibit NFkB through the activation of NRF2. NFkB activates a multitude of inflammatory pathways and induces cytokines that regulate the immune response. One such example is Il-6, which is often activated downstream of NFkB. Higher circulating levels of Il-6 are associated with increased risk for cancers and other age-related diseases. Here, we see a benefit in humans. Healthy individuals given 14 grams of cruciferous vegetables per kilogram body weight daily decrease their circulating levels of Il-6 by 20%. Another study showed that a broccoli sprout powder containing approximately 40 milligrams of sulforaphane, an amount you might get from around 100 grams of fresh broccoli sprouts, reduced TNF-alpha, a marker of inflammation, by 11%, and lowered C-Reactive Protein, another marker of inflammation, by 16% in people with type 2 diabetes. Associative studies in humans show a similar effect on inflammation. When comparing the top 20% consumers of cruciferous vegetables with the bottom 20%, they had, on average, a reduction of circulating IL-6 by 25% and a similar decrease for other important inflammatory cytokines like TNF-alpha, which was lowered by 12.6%. If we accept the premise that sulforaphane, derived from cruciferous vegetables, is driving reductions in inflammation, and this is happening via the NRF2 pathway, then we have to ask ourselves, what is actually happening to NRF2 itself? And what does this mean for other processes inside the body?